माझ्या नानीला मिळेल समज नाही नानीला
थोड़ा समझता मम्मी लेकिन कहती है सफोला वो बहुत इम्पोर्टेंट, हाँ वो बहुत इम्पोर्टेंट है, क्योंकि जैसे मैट्रिक शादी एकदम इम्पोर्टेंट है, तो उसके लिए हम लोग पढ़ते हैं समय से, तो उसमें वो बात सिखाता है कि एग्जाम्स में चलते कॉम्पिटिशन वगैरह, तो उसमें वो मिल जाता है। नहीं ये हो गया, लेकिन पीछे का एक सॉकर तुझे चॉइस गाने दो बार ये करना नहीं चालू है दो बार क्लिक किया ये टाइम कितना बाकी है? टाइम क्या हो गया तो ब्रेक खत्म दोनों कनेक्टेड नहीं वो फैन से बंद करके नीचे या ऊपर रखना चाहिए, तो टेम्परेचर क्या करना चाहिए पता ऑन रखना चाहिए और हाँ वो भी कर सकते हैं, आप क्या करना पड़ता है ऑन करके ना स्विंग मोड हाँ वो फैन ये एसी नहीं है फैन है और ये एसी में अरे यार मुझे भी ठंडी लगती है या पहन के ठंडी लग रही है ये अच्छा फाइट ये और ये होमवर्क देखने में खत्म किया था मैंने टीचर सेकंड हमारा मैथ्स लास्ट मिनट कशाला अरे हमारा सेकंड अगर फोन में तो the state of affairs are really important. मुझे याद नहीं आया थोड़ा सा टाइम टेबल ये वाला तो बस छुट्टी के तो गर्मी छुट्टी तो कर गया मैंने मैथ्स किया तो निकाला तो 12 बजे नहीं उसने खेला तो वो रीजन दिया मैंने समझ यार लेकिन आज मेरा प्रॉब्लम नहीं होगा ना तो वैसे अभी हम लोग ऐसे फिर कवर करना वाला पेपर समझ गए वही हमारा स्टार्टिंग इसमें पानी और तो उसमें पानी की कमी होगी ना तो पानी
Good morning.
Good morning.
So, absorption we were discussing, no? We are almost finishing the chapter. A little bit more is left that we are going to study today. Anything that you want to clarify? What was the last topic we had studied with absorption? We had completed with almost half of the chapter. We had completed with characteristics of the root hairs. We had already completed with properties of the root hairs which allow the process of absorption. Then we had learned certain phenomena like diffusion and imbibition and osmosis. No, all these features we are already done with. Then what else? What else did we learn then? Concept of turgidity, flaccidity. I think that also we had already covered up. Concept of turgidity, flaccidity, and what is meant by turgor pressure and wall pressure and root pressure. No, we had already said that. We are going to continue with this. What are the chapters I'm done here? What are the chapters we have done already? Structure. We are done already with structure of chromosomes and cell division. I'm going to take a sudden 10-mark test on that chapter today. SAQ? Reaction was not good enough. This is what I did not expect. दूसरे बैचेस में क्या होता है पता है? The moment I say this, yes ma'am, हो जाए, no problem. अरे ऐसा एटीट्यूड दिखाओ. You should be very confident, no? भले तुम पढ़ के नहीं आए आज. But this is the way how you test yourself. मुझे कितना आता है देखते. How much you have understood. This mark is not going to get added up in your assessments, in your board exam. But this mark that you get today will make you confident. और will boost up. अगर 1 मिलता है, 2 मिलता है, 4 मिलता है, you should develop that confidence कि मुझे आगे वाला टेस्ट में full marks लाना है. उसके लिए तुम रेगुलर पढ़ोगे, थोड़ा हर दिन. And let me tell you one thing, 10th standard students, you actually have to study a lot as compared to CBSE batches. You know, you have to study a lot. पर क्या उसका ये मतलब है अभी 10th स्टैंडर्ड में आ गए हो तो पूरा 10-12 घंटे पढ़ना है? नो. इफ यू नो द स्मार्ट वे ऑफ स्टडिंग देन 12 घंटा बिल्कुल भी जरूरत नहीं है. Maximum for an ICSE student 4 hours. 4 hours of studying is enough. जब स्कूल और यूटी रहेगा तुमको 4 hours भी बेसिक मिलेगा नहीं. 4 hours also you will not get for studying. But now I think you can still devote 4 hours for studying. Daytime में if you are able to find out 2 hours. Evening if you are able to find out 2 hours. And always take a light and a heavy chapter, light and a heavy subject together in a day. The correct method of studying is 2 subject मुझे पढ़ना है एक दिन में. बट क्या 2 सब्जेक्ट पूरा चैप्टर पढ़ना है? नहीं. आज केमिस्ट्री का 2 टॉपिक्स पढ़ा, मैथ्स का एक हाफ चैप्टर कंप्लीट किया, या जो भी है 10-15 सम्स कंप्लीट किए. बायो का एक चैप्टर के 2 टॉपिक्स पढ़े आज. व्हाट यू रीड शुड बी सॉलिड. मैंने 2 टॉपिक आज पढ़ा, एक्टिव ट्रांसपोर्ट और ऑस्मोसिस. ये 2 टॉपिक आज मैंने पढ़ा. उस पे जितने भी क्वेश्चंस है मैंने सारा देख लिया. नाउ मोर और लेस आई एम थॉरो विद दिस 2 टॉपिक्स. पूरा चैप्टर बाकी है, वो मैं धीरे धीरे पढ़ूंगी. इट्स लाइक दैट यू स्टडी. ऐसा नहीं कि आज मुझे केमिस्ट्री पढ़ना, पूरा चैप्टर लेके बैठे 6 घंटा चला गया, अल्टीमेटली यू स्टडी नथिंग. ऐसा नहीं होना चाहिए. As and when we are almost half of the month, and you have realized already that in this half of the month we are almost through with. By the time your school reopens, we'll be done with another 3 chapters. We are already done with 3 chapters. Today I'll be finishing this. I'll start a new chapter today. So अगर तुम थोड़ा थोड़ा करके पढ़ते नहीं रहोगे, you will start panicking just before a test or an exam. So this is my objective, to make you, ये तुमको बोलते ना, मतलब when you get the prick of the pin every day, if the nurse shows you such a big injection, उससे डर नहीं लगता. ले लो, कितना ब्लड लेना है तुमको, ले लो. It's like that. पढ़ोगे नहीं, कल पढ़ेंगे, परसों पढ़ेंगे, करते करते when you get the alarm ringing in your school, tomorrow is the test or 1 week after is the test, then you start panicking. अरे इतना कुछ मुझे पढ़ना है, and there is a nervousness. When you are confident, you do much better, no? When you are nervous, जो तुम्हें आता है वो भी भूल जाते हो, है कि नहीं? So I always want my students to be very organized. Number 1, you should be thorough with your topics. तो ये एक दिन में तो मैजिक नहीं होगा, थोड़ा पढ़ना पड़ेगा. So in a day, always 2 subjects. This is a basic rule I have followed, and I feel everybody can follow this, and I have got feedback from my students who followed, who organize everything really nicely. This is what from my experience I tell you. हर दिन 2 सब्जेक्ट, 2 टॉपिक, एक का, और 2 टॉपिक एक और सब्जेक्ट, अकॉर्डिंग टू द नीड. अब कल स्कूल में कोई टेस्ट है, ऑब्वियसली यू हैव टू गिव प्रायोरिटी टू दैट चैप्टर, टू दैट सब्जेक्ट, वो अलग है. बट हाउ शुड आई रीड एवरी डे? 4 hours रेगुलर बेसिस में इनफ है. 2 घंटा मैथ्स का ले लो, 2 घंटा केमिस्ट्री ले लो, 2 घंटा SST ले लो, 2 घंटा बायोलॉजी ले लो. ऐसे करके यू शुड प्रोसीड विद ऑल द सब्जेक्ट्स. ऐसे नहीं कि एक वीक भर के हम खाली फिजिक्स और केमिस्ट्री करता रहा, एंड देन मैथ्स और बायोलॉजी और SST और इंग्लिश सब पीछे. Not that. You should get good marks in all subjects, है ना? So पहले हम will try to finish with this chapter. Maybe in the next lecture, I will take a short test of 10 marks, which will be only on cell division and structure of chromosomes. ठीक है ना? So I'm still giving you that 15 minutes का break. If you want to discuss anything, see anything, you can do that. But there's no panic. This is only to give you an idea how confident you are in those chapters. जो नहीं आता है, you will again go home and refer and read. First of all, try solving this question. This is about the concept of Active transfer. I'm giving 5 minutes, but after देखो. Now, this is about. We start with this today. As I said, many more things today we are going to study, but today जो हम पढ़ने वाले हैं वो basically experimental basis रहेगा, and we are going to find out what are some of the concepts we are going to learn. But this क्या हो गया? ये सब बंद है क्या? क्यों? ठंड लग रही है? अरे सब cold blooded हो गया तुम लोग? वो ऑन कर दो ना, फिर इसको बंद रहने दो, वो ऑन कर दो. ठीक है, इसको ऑफ कर दो. नो, तुम यहाँ से देख के यू जस्ट आंसर. दिस इज अ कॉन्सेप्ट ऑन ऑस्मोटिक प्रेशर. Osmotic pressure. Try to understand the experimental setup. The first question says, what happens to the level of water in the beaker after the experimental period of time? 2, what happens to the level of the solution in the funnel? 3, what is the reason? And 4, will there be any change in the taste of the water? Will there be any change in the taste of the water? And 5, name and define the pressure responsible. Diagram, you want to see the upper part? Okay, let me just एक बार diagram देख लो. Can't see? Which one? I should. You want me to read out the questions? First step, what will happen to the level of the water in the beaker? एक क्वेश्चन में पढ़ती हूँ, तुम लिखते जाओ, आंसर लिखते जाओ. Read and remember the concept that you studied. It's based on a particular phenomena that we studied. What will happen to the level of the water in the beaker? That's the first. Second, what will happen to the solution in the funnel? What will happen to the solution in the funnel? What will happen to the solution in the funnel? Yeah. Third, what is the reason for this observation? Whatever you have written for the beaker and the funnel, what is the reason? एक reason लिखना है.
From the observation of the second question.
What will happen to the level of the solution in the funnel?
In that reason.
नहीं, first and second जो भी तुमने लिखा है उसको overall reason क्या है?
For both those questions.
Overall reason क्या है? अरे तुमने उसको भी बंद कर दिया. वो भी बंद है.
नहीं, it's all happening.
Is it working?
Yes.
I don't think so.
Yes, ma'am.
No, working it is. Overall reason is number 3. Will there be any change in the taste of the water in the beaker? 4. Will there be any change in the taste of the water of the beaker? That's question 4. अच्छा, फिगर अगर देख लिया है, I can एनीथिंग नहीं आ रहा है समझ में? बड़ा करो और एक
No, she was asking, is that membrane under solution?
is that membrane?
She is asking what is written.
मैं बताओ आंसर
word
ये
That she is asking if that is labeled membrane.
that is labeled membrane. इसके आगे तुम पूछोगे नहीं कुछ भी कौन सा membrane क्या solution. ज्यादा टाइम नहीं दूंगी just last 2 minutes just 2 minutes more. Last 1 minute. So that's it. 1 more minute चाहिए. Last 1 more minute. इससे ज़्यादा टाइम नहीं मिलेगा. This much time is enough to finish these questions. It is enough to just write one word or one sentence. इससे ज़्यादा कुछ नहीं लिखना है. Just couple of words write. I'll start from that corner. What's your name? Please tell me your names again. I forgot.
Nihari.
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 Nihali.
Tanmay.
Tanmay.
Prashansa. Prashansa.
अरे वाह प्रशंसा. Very good. फिलहाल आज ये 3 नामे मैं याद रखता. Nihali बोलो. First answer बोलो. What happened to the level of the water in the beaker?
The level of water will decrease.
Level of water
Will decrease.
निहाली. और क्या बोला था?
Tanmay.
तन्मय. तन्मय क्या बोलते हो तुम? Is she correct? Is Nihali correct? Level of water in the beaker will decrease. Is she correct? रहने दो है तो correct होगी है ना? You decide. Is the answer correct that she wrote? Diagram देखो. I'll again show you the setup. I will again show you the setup properly. प्रशंसा क्या बोलते हो तुम? What according to you should be the answer for the first question? What will happen to the level of the water in the beaker? It will be same. Can you give me a reason why you say that? Acha.
Level of water will increase. No, decrease. Increase
In the beaker. Level of water in the beaker will
increase.
increase. What do you say?
Increase.
Increase. What do you say?
Increase.
Increase. What do you say?
Increase. Increase.
Increase. Okay, अगर तुम लोग सब increase बोल रहे हो तो शायद सही होगा. क्या पता, मुझे भी नहीं पता इसका आंसर. What will happen to the level of the solution in the funnel?
Decrease.
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 Decrease. आज तुम क्या खा के आए हो? What's your name? सोहम. हाँ, आज सोहम क्या खा के आया ब्रेकफास्ट में? इडली. इसीलिए सब गड़बड़ आंसर दे रहे हो. प्रशंसा, what will happen to the level of the water in the solution in the funnel?
Increase.
Level of the water in the beaker तूने बोला सेम रहेगा, तो funnel कैसे increase decrease हो सकता है? अच्छा, यहाँ पे क्या हो रहा है? सुक्रोज सॉल्यूशन है. बाहर क्या है? वाटर. अच्छा, चलो, I will now jump to the last question. कोई लास्ट क्वेश्चन है, फिफ्थ क्वेश्चन है. What is the process? What is the pressure responsible for this?
Osmotic pressure.
यहाँ पे जो तुमको दिखाई दे रहा है, which process is this showing here? Osmotic pressure. ठीक है ना? तो osmotic pressure में ये जो experimental setup that you get to see, let me just show. Now, osmotic pressure में ये जो setup तुमको दिख रहा है, you are very much understanding in this concept of osmotic pressure. There's a pressure exerted on the solution of the funnel. There's a pressure exerted. यहाँ पे देखो, pressure exerted with the help of this piston and weight. So piston and weight has been applied here, and there's a pressure which has been exerted on the liquid of the on the solution of the funnel. I am the secretary. इडली खा के मत आना सोहम for the biology class. सब गड़बड़ आंसर देते. So now, if there's a pressure exerted, if there's a pressure exerted, never mind, the solution here is more concentrated and the solution here is hypotonic, less concentrated. Should there be adequate movement? Should there be adequate movement of water? What did you learn in the concept of osmotic pressure? What did you learn in the concept of osmotic pressure? ये है हमारा beaker, ये है हमारा thistle funnel. यहाँ पे हमने solution लिया था, यहाँ पे हमने वो membrane लगाया था. Then यहाँ पे हमने वो weight ले लिया. And this is our piston, which is exerting pressure on this membrane, on this wall. So ये piston के through we are allowing adequate pressure to be exerted on this membrane. But this is hypertonic medium. This is hypotonic medium. Normally the water should go this way. यहाँ पे arrow क्या दिखा रहा है? वाटर आके यहाँ पे रूम किया, वाटर इज नॉट एबल टू एंटर। क्यों? सो हम आया अभी याद, अरे तुम्हारे इडली का भी नशा तक गया नहीं ना, अभी याद नहीं आया ना।
ऐसा
नहीं, यहाँ पे कोई कुछ मेंशन ही नहीं है impermeable membrane में, that is not mentioned there. But what did you learn on the concept of osmotic pressure? Even though the concentration is high here, even though the concentration is low here, if there's a pressure exerted, the movement of the water will be almost nil. What was the concept of osmotic pressure? The minimum pressure which is exerted, which prevents the movement of water from the solvent to the solution. याद आया? The minimum pressure, अब यहाँ पे इस तरह से pressure apply हो रहा है ना, so इस तरह से pressure apply हो रहा है, that is the reason water, though यहाँ पे water ज़्यादा है, it's a hypotonic medium, though it is a dilute medium, means the water easily flow. No, the water cannot flow. The water cannot flow. क्यों? Because there is a counteracting pressure this side, no? तो यहाँ पे pressure है, तो that's the reason the water cannot easily flow in. यहाँ पे pressure नहीं होता, immediately the water would have flowed inside in the funnel, and the level of the solution in the funnel would have gone up, and the level of the water in the beaker would have gone down. So if we apply a pressure, क्या ये हो पाएगा ऐसे? No. When we are exerting a pressure with the help of the piston, and there is this liquid which is now in contact with the piston and it is exerting a pressure, ये जो movement है ये negligible होगा या तो almost नहीं होगा. This was the concept of osmotic pressure that we have learned. Even though this is concentrated and this is dilute, the movement of the water molecule will be hindered, the movement of the water molecule will be very slow, the movement of the water molecule will not happen. Q. Because this pressure is not allowing the water to go in. What is this pressure called? Osmotic pressure. What is the definition? Definition is the minimum pressure which prevents the movement of the solvent into the solution. Yaad aaya? Now you write down the answers. Correct your answers. What will happen to the level of the water in the beaker? Will remain same. What will happen to the level of the solution in the funnel? Will remain the same. कुछ भी change नहीं होगा. What is the reason for the observation? What is the reason? Pressure applied on the solution of the funnel with the help of the piston. Since a pressure is applied on the solution of the funnel, since the pressure is applied on the solution of the funnel with the help of the piston, the net movement of solvent into the solution will be zero. Net movement of solvent, net movement of solvent into the solution will be zero. Net movement of solvent into the solution will be zero. Are you following now? Are you understanding? Then now what will be the answer for the fourth? Will there be any change in the taste of the water? No. Will there be a change in the taste of the water? No. Will there be a change in the taste of the water? Not at all. Name and define the pressure. Name and osmotic pressure. Define, क्या बोला अभी मैंने? What did I say? What is the minimum pressure which is responsible to prevent the movement of the solvent into the solution. Solvent into the solution. लिखो. Minimum pressure responsible. Minimum pressure responsible to prevent. Minimum pressure responsible to prevent the movement of solvent molecules. Movement of solvent molecules into the solution. Movement of solvent molecules into the solution. Is this clear now? Okay. So for any question this way, read what the experiment is about. पहले देखो, समझो, then you start answering. So this is also a similar setup. Okay, this is also a similar setup. So these are types of concepts that we have to study. Active transport we have already discussed. By the way of active transport, what does the plant take in?
Minerals.
Minerals. Correct. The plants take in minerals. So now, uses of turgidity we were learning. We had stopped with that topic. Now then, the other day we were studying uses of turgidity, where after the concept of turgidity, plasticity, where you have learned what is the meaning of turgor pressure, you have learned what is the meaning of wall pressure. We were talking about that, what type of benefit does the plant enjoy because of that concept of turgidity? What type of benefit the plant enjoys because of turgidity? So that is what we were talking about, uses of turgidity. Uses of turgidity, that is what we studied. We had already studied 4 uses. We are going to study one more use today, that is the drooping of the mimosa, turgor movement, that is what is known as drooping of mimosa, that is the touch-me-not. And this can also be known as turgor movement because it is completely due to turgor pressure, turgor movements. Now what happens? Now this instance, if I show you, I think all of you have come across a situation sometime. Yes, this touch-me-not plants, if you touch, what happens? This touch-me-not plants, if I say that ये जो plant है and the leaves are more or less arranged this way. So we generally say that these are the touch-me-not plants. So if you touch them here, what happens? The entire leaflet which is spread out starts closing. So this is either we say the drooping of the mimosa. It starts drooping because of the turgor movements. But what actually happens if I talk about what actually happens? This is very common. Quite often you must have seen this. Now we try to understand what is the mechanism, what happens. This particular stem, now if I say that if this is the stem, the magnified form, this stem also has received water by the way of the xylem from the soil. So this has also received water from the soil by the way of the xylem. Yes, this process is same here also. In this plants also the process remains same. Now because of this, if I just talk about this region, yes, this region can be drawn a little bit this way, where as and when they start absorbing water, and when we talk about a situation something like this, when the plants are all flat and broad and spread out, the region where this twig or the petiole meets the stem, or if I say the other way, the junction of the stem and the petiole. Now that the water migration is happening, yes, the water is moving in this direction. Now that the water migration is happening, if I say the other way again, yes, the water is moving in this direction, and if this is the broad stem that is there, and every leaf, they are tight filled with water, turgid condition. Every leaf is tight filled with water. But not only that every leaf is tight filled with water, but the junction also, where the petiole meets the stem, is also filled with water. And this broad region is actually acting as a support for them, firmness for them. What is this region known as when it is tight filled with water? This is known as the pulvinus. This particular region I'm talking about. This particular. But now when we are touching this, what's happening? When we touch this, the movement of water molecule from this starts. The movement of water molecule from this starts. तो ये वाले plant में क्या हुआ, ये वाले leaf में क्या हुआ? This leaf, if I elaborate, it is something like this, say suppose, and it has got a large number of interconnecting cells. The moment you touch, यहाँ पे जो extra amount of water था ना, every cell is more or less turgid in this condition. So यहाँ पे क्या हुआ? The moment we touch, the touch is acting as a stimulus, and the water movement starts taking place. कैसे? From here, the water moves to the next cell, and then to the next cell, and then goes to the stem region, to the the twig region, branch region, and then it will go to the next cell, and then go to the next cell. So यहाँ पे क्या हो रहा है? When the water is getting drained out, this leaf deficiency of water की वजह से it starts almost closing down. तो ये deficiency यहाँ पे build होते होते यहाँ पे भी होगा, क्योंकि the stimulus will move on to the next region. This is the broad leaf that I talk about. तो ये जो सारे cells है, तो ये stimulus यहाँ पे हमने we had touched here, but the stimulus will pass on to the surrounding areas, and the moment the stimulus passes on the surrounding areas, what each of these cells show is there is a panic, and the water molecule starts moving away. The water molecule starts moving in all directions. So this water molecule will start moving in this direction, in this direction, in this direction, sometimes this also. So now when these water molecules are moving, actually what is happening? The cell which is having a large amount of water gives away water to the next, gives away water to the next, gives away water to the next. And that's how the water molecules will also start moving this way. The water molecule will also start moving this way. And then the water molecule will move this way. The water molecule will move this way. So when the water molecule is moving, majorly the moving is upward or in one direction. So when the water molecule is moving, what is happening? Is it the concept of transpiration that we are talking about right now? There is a loss of water? No, the water is not getting lost, but the water is just getting circulated and moving from one leaf to the other, from one leaf to the other one. So जैसे ही water is moving away from one leaf, large number of cells of one leaf, that leaf is getting shrunken. Again from the other leaf, that leaf is getting shrunken. तो ये बहुत fast होता है ये process. The process is very fast. So एक leaf से दूसरा, now see the leaves are straight turgid. जैसे ही इस particular leaf से the water is getting lost or moving, I should not say lost, the water is not getting lost into the atmosphere, but it is that the water is just moving from one cell to the other. The water is just getting moved away from one cell to the other. तो पहले ये swollen region था when it was turgid. Now it has started moving water. The water movement has started, and since the water movement has started, the limping or the shrinking has started. So एक side यहाँ पे देखो in this diagram they are showing that one side has already started shrinkage. Now very soon this side also will shrink. सबसे पहले क्या होता है? When the stimulus is there, the stimulus spreads in all direction. By the time the water movement is happening here, the water movement starts happening from here also to these cells, to these cells, to these cells, to this cell. तो ये region जैसे ही when this region suffers water movement more and the limping happens, this will start hanging because इसका stiffness खत्म हो जाएगा when the turgidity is lost. When these group of cells, what is this part called? Alveolus. So ये जो water movement यहाँ पे तो हो ही रहा है, but the stimulus has gone to this region also. The stimulus has reached this way, the stimulus has reached this way also. So ये stimulus जब तक यहाँ पे भी पहुँच गया, and the water movement in these cells also have started. The cells of this region has started deficiency or has started experiencing deficiency of the water. So when the cells of this region has started experiencing deficiency, then what will happen? ये जो water यहाँ से move होके आगे 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 जा रहा है, these regions are becoming narrowed and grouping down, and the leaves are closing. But you must have noticed, ये जो process of grouping and closing lasts hardly for a couple of minutes, and then again they come back. तो अगर loss of water होता by the way of transpiration, तो ये coming back इतना fast नहीं हो सकता था, और their expansion back or straightening of back इतना fast नहीं हो सकता था. Meaning, when I talk about this, that yes, अभी तो ये grouping हो रहा है, ये closing हो रहा है, but you will see that just in a couple of minutes again they come back to this state. तो ये इतना फास्ट कैसे होता है? How do they come back to the state again? How do they come back to the turgid state again? Because ये water molecule तो loss नहीं हुआ था ना. There was no loss of water happening by the way of transpiration, evaporation. But the water movement was just getting moved from, एक तो इस तरह से move हो रहा था, फिर इस तरह से move हो रहा था, फिर वापस ये tip से आके फिर से they'll start moving, and then again the turgidity will be achieved. एक few minutes के बाद ये turgidity जो पहले loss हुआ था because of the movement of the water this way, now the water is circulating and coming back again. ये circulate होके water when it comes back again, the turgidity is again achieved. So जैसे turgidity फिर से achieve हो जाएगा, the plants, ये जो leaflets है, छोटे-छोटे leaflets, all they'll start opening up again, and this is giving the firm stiffness. So now again they'll open up, again they'll open up. So what are we studying here? We are studying here that ये जो stimulus है, first of all there is a stimulus, and this stimulus will immediately allow the movement of the water. Because of the movement of the water, the turgidity of the cells will be lost gradually. That's the reason we say it's a turbo movement. Why is it a turbo movement? Why it is exhibiting a condition of turbo movement? Because of the movement of the water, the turgid condition of the cell is getting converted to flaccid. अब flaccid condition क्या ये बहुत देर तक रहेगा? No, ये flaccid condition इतना ज़्यादा देर तक नहीं रहेगा because it is just circulation of water. There is no loss of water. Also, if I talk about water being kept in a solution जहाँ पे endosmosis, exosmosis होना, यहाँ पे तो वो भी नहीं है, खाली एक वाटर मॉलिक्यूल दूसरे सेल में जा रहा है, वहां पे सरप्लस जो है वो तीसरे सेल में जा रहा है, वहां पे सरप्लस हो गया, सो दूसरे से अभी तीसरे में, फ्रॉम द थर्ड टू द फोर्थ, फ्रॉम द फोर्थ टू द फिफ्थ, यही एक सर्कुलेशन ऑफ वाटर हो रहा है. Always to understand, is there any loss of water during the process of turgor movements? No, there is no loss of water. How do we understand? What is the proof that there is no loss of water? There is no loss of water, that's why the state gets revived from the flaccid to turgid condition in a very short period of time. Agar loss of water hota, then again osmosis and again cell to cell osmosis and again, this would have taken a long time. Did we see the tulsi plants आजकल by afternoon it gets quite shrunken and dried up? तो क्या ये 2 मिनट में वापस retrieve होता है? No, in 2 minutes it doesn't get retrieved. By evening, late in the evening, you may say, now the leaves have become fresh. तो ये 4-5 घंटे लगते हैं. It takes a long time. क्या इसमें 4-5 घंटे लगता है? No, few minutes. Why? No loss of water. It is just circulation of water and again coming back. Circulation of water and coming back. तो ये ज़्यादातर from the site of stimulus to all directions हो सकता है. Even if you touch here, will this region get affected? Yes, it will get affected because it is just circulation of water from one region to the other. Did you understand now what is the mechanism?
No, but then why is this not happening with other plants?
It is not happening with other plants because other plants have got stiff tissues in the form of well-developed bark, well-developed parenchyma in this type of plants and some others also. The deposition of parenchyma and a specific type of tissue known as sclerenchyma is very less. So there are some few plants like this type of plants are very commonly occurring. But इस तरह के और भी few species of plants है which are very sensitive to stimulus.
Just touch.
Touch. It is not only our touch. It is not only understanding our touch. Even if a bee sits here, the same thing it will show. Even if the lashing of a cow, the same thing it will show. So when we are talking about these are called nastic movements in the plants. So, and mechanism is nothing but turbo movements. The concept of turgidity followed by flaccidity, again turgidity followed by flaccidity. So that is what is happening. So जब तक stimulus नहीं, the water molecules are in their position. जैसे ही stimulus हुआ, the water molecules are mad as if they want to move this way, they want to move that way. That is what is happening inside the plant, inside the leaves. Understood? So this is what we understand as turgor movements. An example that we pick up is the drooping of the mimosa, the Dutchman flag. Now, we already understood the definition of root pressure. कौन बोलेगा root pressure का definition क्या था? इसको याद है root pressure? What was the definition? Root pressure.
From the living cells of root into the xylem.
हाँ, water passes from the root to the xylem. और क्या ऐड करना है उसमें? Root pressure का क्या था? Complete definition. Root pressure. We are going to learn certain concepts of root pressure now. Root pressure. What did I say? What is the definition? The pressure which is developed in the root cells. So ये है मेरा plan, and I'm talking about that these are, when I magnify this region, perhaps I get to see that the root cells are developed this way. And now what's happening? Water entering here. You know now what is this method? This is osmosis. This is osmosis. What did you learn? Tanmay, can you tell me why does water enter into the root cells, root hair cells? What is one major reason you will give me? Why does water constantly, why does the soil water constantly enter into the root hair cells? Because what is the big reason जिसके वजह से the soil water is constantly entering into the root hair cells?
Soil water is hypotonic in nature.
Cell water is, or cell sap is?
Cell swap is hypertonic.
Hypertonic, more concentrated in terms of ions, no? And this medium soil water is hypotonic. सबसे important यही है reason for inward osmosis. Abhi, now yes, the water has come in. So much of water from the outside environment has come inside the cell. Then what will happen? This water will move from one region to another. The water will move from one region to another. So this movement of the water, we already studied that जैसे ही water has come into this cell becomes turgid, water goes here. This cell becomes turgid, water goes here. This cell becomes turgid, water goes here. This is how the water keeps moving. This is what we call it as cell-to-cell osmosis and followed by ascent of sap. Okay? So now you try to understand what is root pressure, the pressure which is developed at the roots due to continuous inward movement of water and cell-to-cell osmosis, which allows ascent of sap or the upward movement of water by the xylem. Pressure which is developed at the root cells due to continuous osmosis and which leads to cell-to-cell osmosis and ascent of sap. तो ये तीन चीज यहाँ पे लिखना है. So now we try to understand very simple that what is the root followed by. Now see here, what is the root followed by the soil water? दिखाई दे रहा है? These are the single root hair cells we had studied in the first day, no? When we started with this chapter, the single root hair cells constantly absorbing water by endosmosis. Now, what is the pathway through which the water moves? कौन से cells है? What is the pathway through which the water moves? Now, if I have to talk about the section of the roots, what is the pathway? See there, the soil water, first of all, will enter through the root hair cell. Then this water will move through the epidermal cells. This is second. So the water, so first of all, if you have to write down a sequence, the soil water, what is the pathway? This pathway you should remember. You get objective questions on this. Options will be all jumbled. You have to select the correct option. Correct sequence तुमको याद रखना है. Soil water. Soil water कैसे? From the soil, the water will go into the root hair cell first. From the root hair cell, where will it go? Epidermal cell. Then from the epidermal cell, where should it go? Cortex. Now, what is the meaning of cortex? Cortex is actually the outer peripheral layer of the cells when we talk about the roots. So, दिखाई दे रहा है यहाँ पे, this is the epidermal layer of cells, followed by this entire region of cells known as the cortical cells. This is known as the cortex. Are you able to see the spherical cells in 2-3 layers? No, they are. Right, start from here to here. This entire region is the cortex. So I would say the soil water enters the root hair cell, moves through the epidermis, then the cortex, then one layer of cell. What is this called? Endodermis. Cortex ho gaya, then endodermis. Then from the endodermis, it will straight away enter into the xylem. Xylem is always in the center of the stem of the root. Leaf may not exactly in the center, but in the stem and the root they are almost in the center. So where now? From the endodermis, where is the water entering? Into the xylem. Will the water be entering into the phloem cells? No. The water will not enter into the phloem cells because we already know that the phloem is responsible for the migration transport of the food. So then, after the cortex, where should it go? Endodermis. And from the endodermis, the water will enter into the xylem. Now, I can also write that it can be the xylem of the root. Let me complete. Xylem of the root. Fifth, xylem of the stem. Then, xylem of the leaf. Xylem of the leaf. And then the same water can be diffused out into the atmosphere by the way of the stomata. Correct, by the way of the stomata. So this is the complete root. Okay? So right from the soil water, right from the soil, how does the water move? I want you to write down, all of you. And this is with reference to the section of the root that we try to study. Sometimes you get this figure in your paper and many questions on this. So पहले तो ये pathway एकदम you should be very confirmed about the pathway. पहले soil water, then root cell, then epidermis, epidermis, then cortex, then endodermis, then the xylem. Now if I have to segregate xylem of the leaf, root, xylem of the stem, xylem of the leaf.
And what is pericycle?
Pericycle is another layer of cells. Pericycle is just after the endodermis, if you see. Pericycle, if you write, it's okay. Majorly we are interested to know this. Pericycle यहाँ पे आएगा. इसके बाद pericycle, इसके बाद xylem. ठीक है? So ये pathway याद रखना है. You get questions on this. देखो, if I am showing you a similar section, this is the xylem. The water has come all the way, and then from here the water has come. No? I'm talking about agar this is the section. The xylem is somewhere, and then from the xylem the water. So if I talk about the section of the leaf, the xylem of this is the xylem of the leaf, and the xylem of the leaf is it connected with the stomata? The xylem of the leaf and the water can be released this way. The xylem of the leaf, and the rest, if I go down, then I get the xylem of the stem and the root and all these cells in a sequence. So this is how we talk about the connection of the movement of the water. Now, when we are talking about root pressure, something very common that we have to try understanding is. अब जब ये root pressure है, yes, we are very much sure that because of the presence of root pressure in the root hair cells, the turgidity is built up in the cells. The turgidity is also built up in the cells of the xylem. and that is also responsible for the stiffness of the plant fiber. Also, you have studied. But now, during the daytime, we will come across something like this. Say suppose now I come across that this is a leaf, and these are the xylem cells, and these xylem cells are getting connected to the twig and the petiole and the stem and the midrib and the cells of the leaf in all directions. No? Now these are the basically cells of the leaf. Now these are the cells of the leaf. These cells are the cells of the leaf. So now what's happening? Yes, the water is moving, upward movement of water is happening. And then from the xylem, the water will go to the cells of the leaf. From the xylem, the water will go to the cells of the leaf. This migration is constantly happening. And all these connected cells of the leaf, wherever there is stomata, now if I say that there are some guard cells here, there are some guard cells here, yes, the loss of the water by the way of the guard cell is happening. Now this process is transpiration. Observe this process is happening, transpiration all throughout the day because the stomata is open. But at night what's happening? This process is closed. This process will not happen at night. Kyun nahi? Because stomata is closed. At night this process will not happen. This process meaning the release of water in the form of vapor by the way of stomata cannot happen at night. But is this process continuing? हाँ, ये process तो continued है because the soil has got so much of water. The soil is well watered. ये process continued हो रहा है. This process is still going on. This process did not stop. But क्या stop हो गया है? In the level of the leaves, on the surface of the leaves, the loss of water by the way of stomata. ये बंद हो चुका है. But now when we talk about evening and night, अब जब ये प्रोसेस बंद हो गया है, is it not every cell is turgid with water? क्योंकि ये प्रोसेस तो कंटिन्यू हो ही रहा है. This process is still continuing. And now if this process is continuing, what else? The plant will have to find out an alternative pathway to deal with the excess of water. Otherwise the cells will start bursting out. Already you have studied when a cell becomes turgid, what type of pressure does it start exerting on the walls? Turgor pressure. But does the cell burst out immediately? No, because there is a counteracting wall pressure. And these two pressures start maintaining a balanced state of the cell for a long time. But now I say, oh, the soil is so well watered, there is so much of water in the soil and this process is still continuing, but the outlet of the water has stopped at night. So now what will happen? The leaf cells, they will start, because they are constantly receiving water. Now the leaf cells, they will start giving away water through other pores which are there on the surface of the leaf. Because stomata to bhi band hai. But they have to find out a way by which they have to give away the excess water. So now what will happen? There are some specialized pores on the margins of the leaf apart from the stomata. Apart from the stomata, there are some small pores on the margins of the leaves, and these small pores are called hydrathodes. These small pores are called hydrathodes. So ये जो excess water, whatever the cell has gathered, the water will come out in the form of a small droplet. The water will come out in the form of a small droplet. At what time of the day do we generally see this? At night, early morning, where the temperature is very less, intensity of sunlight is not favorable for the process of stomata to open. Intensity of the light is not favorable for the process of photosynthesis to start at that time. Do we see these droplets now? No, never. Abhi kyun nahi? Because now the stomata is open. Intensity of sunlight is favorable for the process of stomata to open and photosynthesis to run. So अब जब stomata is open, these hydrathodes, they will not be dealing with releasing the excess water because stomata is doing that job. So this process by which the leaves give away excess water in the form of droplets, this process by which the leaves give away excess water in the form of droplets is what is known as guttation. What is guttation? The process by which leaves give away excess water in the form of droplets. When can this happen? Majorly in the evening or at night when the stomata is closed. When the intensity of sunlight and all these things, temperature, etc., are not favorable for photosynthesis, are not favorable for opening up of the stomata at that time. Early morning, midnight, early morning, late in the night, this process would happen. What is the other condition that should be there? Yes, the soil should be well watered. If the soil is dry, early morning, are we able to see guttation? No. Soil is dry, then the cells are as it is running short of water. So the guttation ka koi sabal nahi. So that is what we are talking about. What is guttation? Guttation is a release of water in the form of droplets through specialized pores on the margins of the leaf, known as hydrothodes. Known as hydrothodes. तो अभी इसका रूट प्रेशर से क्या लिंक है? रूट प्रेशर से यही लिंक है because the cells are root cells are constantly taking water from the soil because the soil is in a hypotonic state. Since the soil is in a hypotonic state, so much of water is still entering. Cell to cell osmosis has been allowed. So ये जो पूरा process इसके लिए कौन सा pressure responsible है? Root pressure. So already we had studied the pressure responsible for the continuous inward movement of the water into the root cells, which allows cell to cell osmosis and upward movement. That is the root pressure. तो अभी root pressure की वजह से ही hydrothodes से excess water will be released out. Whichever part of the day we have learned, the excess water is released out only because of this pressure. Where is it acting? At the root cells. It is acting at the level of the root cells, hydrothodes. Now, we are going to learn some other concepts of ascent of sap. Ascent of sap already we have studied. What is ascent of sap? Upward migration, upward movement of the water through the cells of the xylem. Which process takes place during ascent of sap itself is a process, but name the process responsible for ascent of sap. क्या लिखना है?
Cell to cell
Osmosis. Cell to cell osmosis. ठीक है? So एक cell turgid है तो surplus water दूसरे cell में जाना है. That is cell to cell osmosis. Now let us learn some experiments based on this concept of ascent of sap. बहुत simple experiment है. देखो, try to understand. We are talking about two test tubes, P and Q. ठीक है? In both the test tubes, what is common? We have taken almost 3/4 filled with water, and then we have taken a shoot. The shoot is having well-developed roots in one. तो एक टेस्ट ट्यूब में we have taken well-developed shoot जिसके roots भी intact होने चाहिए. Enough amount of roots should be there. A small shoot with leaves, well-formed shoot होनी चाहिए, and put it in the test tube. And then in both the test tube, add few drops of oil. And let it be there for few hours. After few hours, what are we going to observe? After few hours, we will observe in the first test tube, that is the P that I call it as P. In this test tube, will there be any drop in the level of water? Yes. If I talk about 5 to 8 hours, 6 hours, हाँ, drop तो रहेगा पर थोड़ा सा, ज़्यादा नहीं, थोड़ा सा. इसमें कुछ drop रहेगा? Nothing. Almost nothing. इसमें भी कोई remarkable drop नहीं होगा, but फिर भी होगा. अब ऐसा क्यों? Because when I talk about this test tube, there is a layer of oil here, otherwise there is water all over. And then now I talk about the leaf जो है, the shoot that is there, which has got well-developed roots, and then the leaves. So what is happening? Why there is a drop in the level of the water? Because since it has got adequate amount of roots, The water will first of all enter into the root cells. The water will move by the way of the xylem, and now that the water reaches the leaf cells, adequate amount of water will be lost also. And since this amount of water is lost, that is what is responsible for the intake of the water also. जितना ज़्यादा loss, उतना fast intake. जितना ज़्यादा loss, उतना fast intake. So if I talk about a bright sunny day, will this process be fast or slow? Fast. And if I talk about a gloomy day, will this slower? The process will be slower. You are able to find out the connections now?
Yes.
So this is associated with ascent of sap. So अभी इसमें क्यों कोई change नहीं हुआ? क्योंकि हमने oil दे दिया था. तो surface of the water undergoing the process of evaporation वो तो हमने stop कर दिया था. Because we had applied small amount of oil, तो oil की वजह से surface evaporation नहीं हो पाया. Otherwise इसमें भी drop होता था. अगर हम oil नहीं देते, then surface evaporation की वजह से इसमें भी drop in the water level इसमें भी दिखता था. But अभी in the second test tube there is no drop क्योंकि surface evaporation had been stopped by applying oil. You get questions which is acting as a control experiment किसे कहते हैं ना? In this sort of experiments you have to remember that word. Control experiment is one जिसमें कोई observation तुमको remarkable नहीं दिखेगा after the period of time. The setup which does not show any observation. The setup which remains same after the experimental period of time. The setup जिसमें कोई remarkable change तुमको नहीं दिखेगा after the experimental period of time. That type of a setup is what is known as control setup. But क्या इसमें कोई remarkable change दिखाई दे रहा था? हाँ, at least water level अगर by 0.5 millimeter भी अगर कम हुआ है तो कम हुआ है ना. But इसमें उतना भी तो कम नहीं हुआ, इसमें तो उतना भी change नहीं हुआ ना. So that is what we are talking about. That the change that takes place after the experimental period of time, we call it as observation. और जिसमें कोई observation नहीं है, we call it as control. तो ऐसा क्यों हम control क्यों लेते हैं? To find a comparison. Otherwise हम कैसे compare करेंगे? To find a comparison. अभी can you tell me? ये तो एक हो गया by applying oil and not using the shoot. We stop any observation. यहाँ पे कोई observation इसीलिए नहीं है क्योंकि एक तो हमने oil दे दिया and we did not apply any shoot. We did not keep any shoot. What can be the other way जहाँ पे we see that कोई observation remarkable नहीं रहेगा? What can be the question you get is what is the other way of setting up a control? What can be the other way of setting up a control जहाँ पे तुमको experimental time के बाद में कोई observation नहीं मिलेगा? What can be the other one? Think.
Ma'am, in this experiment, we see that in the container P, which is not a control setup, there is the balancer plant. But then in the container Q, we only have the water and the oil added. And so we can compare.
ये तो हो गया control. I want to learn what can be the other way of setting up a control जहाँ पे कोई observation नहीं हो. One way of setting up a control is take a test tube, fill with water, put oil, don't keep shoot. ये एक control हो गया. What is the other way of taking a control? Control का मतलब हमेशा ये याद रखना है 5 hours, 10 hours कोई ज़्यादा remarkable observation नहीं होगा. कैसे हो सकता है एक बार?
Not putting water and putting the shoot.
Not putting water, putting the shoot. Not exactly.
By using a dead plant.
Dead plant. by putting a dead plant. अगर तुम dead plant use करोगे, the shoots are or the cells are not going to show any absorption. I take यहाँ पे what I do is maybe I have not taken oil. मैंने यहाँ पे oil नहीं लिया. I took this live shoot, water, no oil, water and dead shoot. तो हमारा next setup में क्या है? We did not take water. I'm sorry, oil. Oil नहीं डाला दोनों में. एक टेस्ट ट्यूब में वाटर है, शूट है. दूसरे टेस्ट ट्यूब में वाटर है, डेड शूट है. Then will the second test tube act as control?
Yes.
Yes. तब भी second test tube में कोई change नहीं आएगा. And first test tube में तो अब remarkable change आएगा क्योंकि हमने oil नहीं डाला. Evaporation भी होगा और transpiration भी होगा, दोनों होगा parallel. So in that case, if you don't put oil, water level will drop remarkably. पर यहाँ पे फिर भी कोई ज़्यादा change नहीं होगा, थोड़ा बहुत खाली surface evaporation जो होना है वो होगा, that much only. So that is what we talk about as ascent of sap. Now this is the second experiment. What is happening here in the second experiment? Again, we take a beaker half filled with a colored solution. See this, you have this picture in your books. Here. देखा? You have this experiment. ठीक है ना? ठीक है? Now let us see what's happening here. Let us see what's happening here. So इसका setup क्या है? इसका setup is something like this. What are we doing? We are taking a beaker. This beaker is half filled with a stained solution. Eosin. We take a stained eosin. So now, when we are talking about this experiment, what is happening? We are taking a beaker. This beaker is filled with a colored solution. We basically take the water. We just drop some. We add few drops of colored solution known as eosin, and then we take our plant. We take a shoot, but the shoot should have adequate number of roots and leaves. सब होना चाहिए. Next, what are we expecting? We are expecting that the water will enter into the roots. The water will start migrating by the way of the stem, and the water will enter into the leaf cells also. Are we able to understand that the colored water is moving into the different parts? The leaf is green. Are we able to see any color? No. The stem is almost brown. Are we able to see any color from the outside? No. The roots are brown. Are we able to see any color from outside? No. So we want to understand that yes, the colored water. Now, what is the need for taking the colored water? क्योंकि हमें ये route of migration of the water समझना है. We want to see in what particular route, in what particular pathway, the water kept on moving from the root to the stem and the leaf. ये हमें समझना है. अभी we cannot see the color from outside. Color तो नहीं दिख रहा है. Here they are showing that it's red. Actually, it does not appear. No color change. You keep this plant in the colored solution for almost 8 hours. क्या तुमको बाहर से roots red दिखेगा? No. क्या तुमको stem red दिखने लगेगा? Nothing. Leaves also will remain green only. Color इतना prominent कुछ भी नहीं दिखेगा. But now we want to see तो कैसे करेंगे? We have to take a section of the leaf after the experimental period of time, some 6 to 8 hours, 10 hours, 12 hours, जो भी हम decide करें. Take a section of the stem, take a section of the root. Take a section of the root, take a section of the stem, take a section of the leaf, and we should more or less have the idea कि एक leaf के section में xylem cells कहाँ पे placed होते हैं. एक रूट का सेक्शन में xylem cells कहाँ पे प्लेस होते हैं. I gave a fair idea. अभी जो हम डायरेक्शन ऑफ द वाटर सीख रहे थे, तो what was the migration route? soil, root hair cell, epidermis, cortex, endodermis, pericycle, xylem. ये पाथवे हमने सीखा है ऑलरेडी, बट हमें इसकी नॉलेज होना चाहिए कि जाइलम अगर रूट है तो कहाँ पे सेक्शन में कहाँ पे प्लेस्ड है वो सेल्स, स्टेम है तो सेक्शन में कहाँ पे प्लेस्ड होना चाहिए, दैट इज द सेक्शनल व्यू वी शुड मोर और लेस हैव एन आइडिया. लीफ है तो कहाँ पे प्लेस्ड है. तो सेक्शन जब हमने काटा उसके बाद हमने ये देखा कि if it is the section of the root, then the central region of the cell is getting stained with that color. The central region of the cell, surrounding cells, no, none of the cells, the surrounding cells में किसी में भी वो colored stain आया ही नहीं. But it is only the central region of cells which have taken up that stain, which became colored little bit. अच्छा, next stem वाला section में क्या हुआ? Stem वाला section में क्या हुआ? In the stem we come across not all cells of the center had picked up the stain, but छोटे-छोटे regions of the cells. So when I talk about the section of the stem, in the inner line, some cells, and the arrangement of the cell is something this way. In a circle, bit away from the center. In the section of the leaf, what has happened? Bit almost in the center, but only a small group of cells, surrounding cells are not affected by that stain. Later it is found out that these cells and this group of cells, they are connected. And these group of cells and these red colored cells, they are connected. And this is what is the xylem cells. The different group of cells of the xylem, the 4 types of cells of the xylem, they remain arranged in different way in the root and the stem and the leaf. And these cells which are colored are the xylem cells of the root. These cells which are colored in the stem are the xylem cells of the stem. The xylem of the stem is arranged in a different way. And these cells which are colored in the leaf are the xylem cells of the leaf. So what do we conclude? Yes, this colored water which has been absorbed and which started moving up is all the xylem cells only. No other cells have been involved in the upward migration of the water. It is only the xylem cells which are helping in the upward migration of the water. From the root to the stem to the leaf, it is only the group of xylem cells which have been continuing their process of ascent of sap. So this is what you understand from this experiment followed by this observation. Is this clear?
Yes.
Anything that you want to ask? Any doubt? Any doubt in this? So we learned 2 experiments. One is that simple oil experiment. And second is the eosin or the stain colored water experiment. In both this, what did we learn? Ascent of sap by the xylem. Upward migration of water by the xylem. Both the experiment objective is same. Now we talk about the third condition. What is this setup about and what does it explain? This also explains water is conducted by the xylem. So many different ways it has been proved that yes, it is the xylem tissue only which is allowing the upward migration of the water. तो यहाँ पे क्या हो रहा है? यहाँ पे देखो, we take 2 tissues first of all. We take 2 tissues. कैसे? This is a stem region. We take 2 stem region. Should there be intact leaves? हाँ, intact leaves दोनों में होनी चाहिए. Intact leaves दोनों में होना चाहिए. It should be there. Intact leaves should be there. So now, in one setup, what has been done? In one setup, the central region, in one setup, it has been, an incision has been made. Wait, I'll show you. In one setup, an incision had been made this way, in such a way that in one setup, the incision had been made in such a way that the outer region, what I told you with this concept, that whenever I talk about, again remember, I'm talking about a stem majorly. Stem में, the xylem is always in the center and the phloem is in the surrounding region of the xylem. The phloem is very close to the bark. So ये है phloem, ये है xylem, ये है phloem. This is the setup of the vascular tissues in the stem. So what did we do? Which tissues did we remove?
Phloem.
We removed the phloem. We kept the xylem intact. We did not disturb the xylem in this tissue. यहाँ पे क्या किया? यहाँ पे एक central incision काट लिया. तो what did we keep intact? Phloem को intact रखा है and xylem को निकाल दिया. आ रहा है समझ में?
Yes.
And then बाकी we kept it in a dilute hypotonic medium में ही रखना है. So attach that with the clamp and stem, that's all there. So in the first case, what did we remove? We kept the xylem, removed the phloem. Second में we kept the phloem, removed the xylem. ठीक है? And we keep it for a particular period of time. अभी for a particular period of time when we keep, ये water है दोनों में. Which leaves start wilting and drooping? This leaves start wilting and drooping. तो ये वाले leaves का क्या difference हो जाएगा? They will start the process of wilting. They will not be stiff after a particular period of time. They will start the process of wilting and drooping. क्यों? Because the inward movement of water हो रहा था. The water was moving, but यहाँ पे जाके water movement interrupt हो गया. The water could not go to the upper part. Can the water go? Can the water go to the upper region? No, the water cannot go to the upper region. Since the water cannot go to the upper region, the water movement has stopped gradually. Since the water movement has stopped gradually, these leaves had started losing their turgidity and became flaccid. And hence the drooping. यहाँ पे क्यों drooping नहीं हुआ?
Xylem is still intact.
The xylem is still intact. तो यहाँ पे जो भी water आ रहा था, the water movement kept on happening, the water movement kept on happening, the water movement kept on happening. कोई problem नहीं थी. And all these leaf cells got adequate amount of water. So after that experimental period of time, these leaves are still turgid. They are spread out. But to conclude, xylem cells are necessary. Xylem tissues are responsible for the upward movement of water. Xylem tissues are responsible for the ascent of sap. है? So this is the third experiment that we talk about. What about this? Now we try understanding one more. This condition, quite a number of times you will get to see in these plants also, these big plants. Quite often when we walk through the road, the big plants on the sides of the road, very common we get to see this. काफी बार, if at all you had observed that these big plants with their broad trunks, quite often they show a swelling. They show a swelling like this, a swelling like this. क्यों? अच्छा, what is this setup? अभी, what is happening here? In this setup, what are they doing is make a narrow cut here. इस तरह से select a stem of a well-developed plant, shoot, जो भी है, select it, and maybe now this is in the soil. Let me just talk about a regular situation also. It is in the soil. अभी क्या हो रहा है? Now Maybe now these are leaves attached to this upper part. What process is happening in the leaves? Photosynthesis. Food is getting formed. The food is getting distributed this direction, and even this direction, and even this direction, and even this direction, and even this direction. But can the food come down here? No. The food cannot, I'm sorry. The food cannot, this is xylem, no? The central region is xylem. So since this central region is the xylem tissues that I said, so kaun sa tissue had been cut out? Phloem had been cut out. So the food which is supposed to get migrated by the way of the phloem, can this downward migration happen? No, the downward migration ho nahi raha hai because we had taken out almost about 3 to 4 centimeter of this much length, or even a little bit more, 5 to 6 centimeter of this phloem, of this bark also. If you just scrap out, cut out, the continuity of the food movement will stop. What do you expect for a leaf here? Will it remain fresh, or it will wilt or droop? What do you expect for a leaf here? Huh? It will be fresh. Acha. The food movement is not happening. The food movement has stopped. Since the food movement has stopped, will the leaves in the lower region of the stem get affected? Not much. Kyun? Because water movement to ho raha hai, and these leaves also will undergo the process of photosynthesis on their own. But will there be any change in the upper part? Upper part में ये problem होगा कि after some time we will see that these regions have started swelling up because of the accumulation of large amount of food, and the cells are swollen with food. The cells of this region are swollen with excess food. क्योंकि ये food तो circulate and distribute नहीं हो पा रहा. But for time being, these leaves will be able to prepare own food because they are getting the water. But after some time, this region of the stem also will show some dryness. These leaves also may get affected because the regular supply and circulation of the food is not there. And the upper region of the stem progressively starts showing this swelling, and this is what is known as girdling. This is what is known as girdling. This is what is known as girdling. Why does girdling happen? Is girdling happening in the lower region of the stem? No, in the upper region of the stem. Because food is coming from the green parts of the leaf downwards. तो वो downward migration का path हमने रोक दिया. You're not allowing the movement of the food in all directions to happen. So initially, lower region of the stem will not be affected that much. The leaves will be more or less fresh and the stem. But yes, after some time, of course, after a couple of weeks, yes, after a couple of months, yes, it will get affected because the majority of the leaves are always on the upper part of the plant. The food manufacturing is more on the upper part of the plant. And that food is not able to come to the lower region. So is it not? The lower region of the stem and the roots will start becoming weaker, of course. After a particular period of time, yes. And जो भी leaves नीचे है, they will undergo the process of photosynthesis, but they may not be enough and sufficient for the rest of these parts to support. यहाँ पे अगर 10 leaves है, and ऊपर अगर 100 leaves है, will these 10 leaves be able to support all these regions of the? No, maybe not. So that is what is the concept of girdling. Why does girdling happen? Due to accumulation of food. Which tissues are affected? Which tissues are responsible for the transport of the food?
Phloem.
Phloem tissues. So phloem is basically responsible for the transportation of the food. So this is what we were discussing about the, see here, the same thing, the swelling up of the tissues in an experimental setup if you try. naturally we get to see this. Quite often you will come across, sometimes when normally also when the phloem tissues are getting died out in the plant cells, you will get to see the big plants with big trunks, the lower region of the stem is getting swollen. The lower region of the stem may get swollen, or sometimes the middle region of the trunk getting swollen. And the lower region of the stem appears a little bit narrow as compared to the upper part. That's because of girdling, that's because of accumulation of the food. Take a break and we'll continue again after the break. Are tired? Huh? Yes, ma'am. क्यों? Huh? You're tired?
We were looking at the screen like this, so.
इसीलिए तो आपसे कुछ दिखाऊंगी नहीं, जाओ. चलो, relax, go out also, have some fresh air, थोड़ा घूम के आओ, but चिप्स कोल्ड ड्रिंक्स नहीं लाना है, ऐसे खाली घूम के आओ, stand there outside, बिल्कुल मत बैठो, 15 minutes relax.